Hello and welcome back to our fall installment of Manure Mondays on 2021. This Monday, we are talking about manure management and antibiotic resistance. Adina Howe was going to help me out with this presentation, but she gets to be on vacation this week. So she helped me out with some slides and some thoughts to share with everyone, but you have to make do with me as we go through this thing today. So big thanks to all of you joining us. And if you can't be here, hopefully you'll take a moment and listen to it later after it gets posted online. So what is antimicrobial resistance? It's often shortened to AMR. So if you see that floating around in some abbreviations on slides, it means the same thing, antimicrobial resistance. And all we're really saying is it's the ability of a microorganism to resist the effects of any antimicrobial medicine or anything that we traditionally use to try and treat infections or bacteria. So some of these can be natural things. Oftentimes we talk about copper or zinc being antimicrobial elements or additives that we might use for things. More specifically though, generally when we talk about antimicrobial resistance, we tend to be talking about it in terms of drugs that are generally used to treat infections or human health conditions that have some overlap with either natural immunity occurring in bacteria or because of practices that we're using on a farm, maybe the occurrence of different bacteria that have those genes in them that help them resist some of the treatment methods that we use. There are two types of transfer of resistant genes. And I have some pictures here to help understand them. The one on the left is sort of what we think of probably more traditionally, how antibiotic resistance occurs, right? We start out with a population of bacteria. For some reason, one mutates, it gets this special yellow strand of DNA that makes it resistant to whatever antibiotic we're talking about specifically, and it passes that hereditary gene down to its offspring as it splits and divides. So once it's established in that bacteria, it's a way for it to proliferate. The other method of obtaining antimicrobial resistance, and probably one that we think of at least less traditionally, is this horizontal transfer. Essentially, two bacteria link up for a short period of time, and one injects or donates a gene over to the other bacteria. So we can see here, it has that magical yellow strand that we said was making it resistant to antibiotics, and it can move that gene over to the other one. And this can be an active process where the gene is injected, or in some cases, there's some evidence to believe that that gene starts floating around in soil as different things were decaying, and it gets picked up by a bacteria sort of randomly, and it still assimilates it into its DNA. And the reason that works is because they're stored in special plasmids or things like that that bacteria can just pick up and put down. So when we talk about how does antibiotic resistance happen, it's something that we think of is there naturally, right? There's lots of germs, there's lots of different bacteria out there, and we might think of most of them as these orange bacteria, but every now and then there's a special blue one. If we treat something, we might be able to kill all those orange ones, and that one that manages to survive for whatever reason starts to proliferate because all its competition has been killed off, right? Because we treated it. Some bacteria can give their drug resistance to different bacteria then. Another place that we have tended to see this and started to talk about this in agriculture is with Roundup resistant weeds, right? Especially Roundup resistant ragweed, some of the other ones. We've been using Roundup for a while. Most of the time it does a really nice job of cleaning out our field, making sure that nothing's left viable, but every now and then we get a weed that escapes, and it tends to be that those weeds that escape are more resistant to Roundup. So over time, as they start to become the ones that proliferate, because they're the ones that manage to live through that treatment, we build up this greater and greater tolerance to Roundup. With antibiotic resistance, we tend to have the same thing happening. When we do a normal treatment, we kill off most of them, but the ones that manage to survive, the ones that are more hardy, maybe that happened because they just were already hardy enough to survive, or maybe it happened because our dosage wasn't quite right and we had some escape, but they tend to be selecting for the ones that have just a little bit more tolerance. And since they proliferate, they get to be the ones that pass on their genes. When we talk about how antibiotic resistance spreads, I want to say that there's a lot of places that it comes from. And while we're going to take a manure management focus today, right? So we're going to be talking about livestock production and manure. It is by no means the only source. It's an important source, but certainly when we think about human medicine and human treatment, that's one place it can come from. When we think about antimicrobial soaps that we're all using, a lot of human treatments for keeping things hygienic and clean, right? Those are also potential sources for where they could come from. So while the rest of this presentation is going to be entirely focused on manure, it certainly isn't the only source out there. One other note when we talk about livestock agriculture, certainly as soon as it's excreted, that gene is present in the manure, and we'll look at some treatments and effects that they could have. But for the most part, we're more concerned when that manure leaves its storage and goes out into the environment, because that's where it has a chance to interact, move either into waterways, into soils, into places where it can interact with other bacteria that we might come in contact with. When it's in the manure storage pit, certainly it's there. There's potential risk, but since it isn't interacting with the environment, as long as our manure system's working well, that risk of it moving, of it interacting with people is extremely low. So the first paper we're going to look at is some work done by Adina and Michelle on seasonal variations in export of antimicrobial resistant genes from agricultural watersheds. And the watershed they were working at is Black Hawk Lake watershed over in west central Iowa. They have a nice map for us showing us sort of where that county is, but also things that are happening in that watershed. So anywhere you see a brown field, that's a field that is in a manure management plan and known to have received manure. If you see a tan field, that was a past field that was in a manure management plan but isn't in a current manure management plan. And then the gray squares and gray triangles are hog confinements, and then cow, either beef or dairy, confinements or feedlots respectively. So it is a watershed that has a fair amount of livestock in it. Certainly it's not dominated by livestock, but somewhere around 30–40% of that watershed generally has manure associated things with it. So what they were really interested in is understanding, when do the genes that we typically associate with antibiotic resistance, when do they move? When do we find them in tile or stream water throughout the watershed? So they had some monitoring points, roughly 6 in this watershed, where they'd go take samples on a weekly basis and measure the prevalence of both those genes. And then by plating them, so they'd take that water sample, put a small portion of it on a little media plate where they have some nutrients for bacteria, and then that same plate with different antibiotics on it, either tetracycline or tylosin, to see if any of the bacteria that they were capturing in their water sample were resistant to those genes. And then they'd grow them out and count how many colonies formed, so how many of those bacteria were really there. So this is what their data generally look like. And the ENT is a type of bacteria, right? So it was the specific bacteria that they were plating for. And then they looked at resistance for 2 different things, either TET, so tetracycline, or tylosin, right? So TYL. So TET there, think tetracycline, that's something that we often associate with pig production, especially certain phases of the ration, as an antibiotic, and tylosin, the same thing. And in case you were curious, not as many people use tylosin right now as they used to. More people use thymucin, but the impact on the bacteria, essentially the bacteria and the gene that we look for, are very similar. So they broke the growing season up into 3 phases, either pre-planting, so early spring when maybe some manure application is occurring, that growing season, so shortly after manure application, and when more of our nutrient movement, more of our bacteria movement could potentially take place, right? Because we're getting more hydraulic events. And then that post-season window in the fall, where we tend to see that fall manure going on. They didn't sample in the winter. Some of their sampling points were tile lines, so those tended to either have no flow or very, very low flow in the winter. So they didn't sample then, and then they came back out in the spring. So the big takeaway here is that you can see a large difference in the seasonality of when that manure or when that bacteria was moving. So again, a couple things to keep in mind: manure is not the only source of bacteria in the environment. They can be spread through seepages from manure storages. They could be spread from wildlife excrement. If we had cattle directly in the stream, they could come from there. And they could also come from any human system, right? So if we had compromised or maybe not as effective of septic system as we were hoping for, either for towns or for people, we should see a slow leakage of maybe antibiotic resistant genes from those. In most of those other cases, if it was a compromised septic system or a leaking manure storage, right, were causing the impact, we tend to not associate those with seasonality. They'd sort of be constant with time sources. With wildlife excrement, certainly there's a chance for that. With direct stream contact, we could see some of that seasonality as the wildlife is more active in the spring, maybe out in the stream. The same with pastured cattle. But based on the observations they made, they think that it looks to be most closely associated with that manure application. We see that larger post-harvest number than we see in the spring. That would tend to be indicative of when more of our manure is going on. And then we do tend to see it more in the growing season when there's greater hydraulic movement. Just because it's sometimes really messy and difficult to do some of these studies in the real and natural environment. There's a lot of things going on. Michelle and Adina, and this one's one of Adina's student studies, they've tried to do a fair amount of work here in the lab to better understand these. So what they did is they went up to Nashua, Iowa, took some soil columns, either from plots that had received manure for the previous 10 years or from plots that had never received manure, right? So then they had these two different treatments, and what they're going to do is some rainfall simulators. So they're 6-inch PVC columns, roughly 50 centimeters long, so a good size column, and then periodically they're going to run rainwater through that column, either with manure being applied previously or without manure being applied previously, looking at what's moving through the soil, right? So what moves into that leachate water, similar to what would make it into a tile line. They did do some destructive sampling where they basically took down the column to try and see what bacteria were there. And then they did do sampling from manure to get an indicator of what bacteria were present. So this study is relatively interesting because some of these columns have a manure history, some don't, which can make it a little bit different in how the bacteria started with. So this graph might be a little confusing at first, especially for people who like units on their graphs, because the first thing you might notice is that both the X and the Y axis on this graph are non-dimensional. This is a graph that I had to get comfortable with in the last few years. Essentially, what they're doing is they are performing a census of the microbes that are out there. So they'll count both the types and the number. And they do that by looking for different fractions of genes. And then once they've done that, they find a way to plot all those data points using non-dimensional scaling. And the important thing to remember is that if data points are located close together, those microbial communities are really similar. If the data points are located further apart in distance, those microbial communities are relatively different. And then they have a key up here that tells us what the samples are. So we can see these 4 dots really close together. Those are directly from the manure, right? It makes sense that all those should be relatively similar. They got one manure source. They broke it up into 4 treatments that were going to be applied to their columns and said, what do those microbial communities look like? And they found out they were relatively similar. So a few things. They did do some testing to see what antibiotics were present in their manure. They measured tetracycline, oxytetracycline, and thymucin, tylosin, and monensin, and they did find all of them present. The one that was present at the highest concentration was tetracycline at about 5,000 nanograms per gram of dry weight. So when we report antibiotics in manure, just like you might see on some of your manure tests, it's often reported on a dry weight basis rather than on a per gallon basis. But all these antibiotics were already present in the manure, not too surprising, but the concentrations do vary relatively widely from farm to farm. But they were interested in, are these things causing genes that would move through the soil? So in this case, they were looking for a site that had a known history of using antibiotics and actually sampled the manure from a phase shortly after the antibiotics were used to try and get a larger presence of the antibiotic resistant bacteria they were looking for. So a few other things on here. They did find differences in the microbial community, and this microbial community that they're plotting is actually one related specifically to antibiotic resistant genes. If you look through some databases, you can find there are certain genes that we tend to think of as indicating antibiotic resistance, and those are the ones that they were targeting in their analyses here. So when we look at effluent, that blue triangle, that would be effluent from a column that neither received manure nor had a history of manure application. So what we want to compare is that to that yellow. sort of manure treated effluent, right? So that'd be in columns where we had manure, and then the rainfall event moved some of those manure nutrients and the bacteria associated with that manure down through the column and out into the effluent. And one thing you can see is that there is a difference between the effluent from locations that never had manure and from those plots that both got fresh manure and that got had a manure history. When you see these four dots really close together, those tend to be replicates. So from one point of time, they did see a trend. So as they went through this study and did periodic leaching, the first sample most correlated or most represented that manure, right? They're relatively close together. There's some differences, but directionally, they're still reasonably close together. And with time, we saw more divergence between that manure indicator in that effluent that drained through the soil and what was actually making it out. So that manure signal, the antibiotic resistance that came from the manure was still there, but it did decrease pretty drastically with time. The same thing happened with the soil, right? So occasionally they would take some of their soil cores, break them down and see how many antibiotic resistant genes they could still find in that soil. Shortly after application tends to be grouped up here. And with time, they tended to decrease. So they were more similar to what was coming out. Again, we can see a difference between those soils that had a manure history and that soil that had never received manure. One big thing you can probably note reasonably clearly on this line is whatever was coming out in the effluent tended to be similar to what they measured in the soil, right? So when we started out the study, the effluent that drained through the soil tended to have a similar microbial community to what we found in the soil. And as we went with time, the same thing. The microbial community in the soil shifted, but what was coming out in that effluent that drained through also shifted. I will note that they didn't find that the changes with time were statistically significant, but there is a pretty clear trend that some changes were happening. I wanted to look at one other study on that where they use soil columns, and this study has a name that I really liked. It said, "Do manure-borne indicators of soil microorganisms influence the spread of antibiotic-resistant genes in manured soil?" Right. So if we put this stuff on, do we have a bigger chance of finding it? And this study did something that I thought was relatively unique. So right here on the left, you can see this is just soil. They didn't do anything to it. They didn't add manure. And that was how many antimicrobial resistant, antibiotic resistant genes they found in their soil. And then they took soil and said, we're going to add some manure to it, but before we add that manure, we're going to irradiate it. So we're basically going to kill all the bacteria in it. They took that manure, added it with nothing alive, and said, lo and behold, it doesn't really change how many bacteria we see, right? And the other thing to note is this is incubation time. So how many days into the study we are. So 0, 10, 20, 30, and 60 there. The next one, they said, what happens if we irradiate the soil and then add manure? And they got this purple curve. So right after they added the manure, we saw a large number of antibiotic resistant genes present in that soil that did decrease during time. And the one that we all probably think of as the most common would be what happens if we just have soil and we add some manure and we don't irradiate either before we do it. And that's that green line. So and this correlates relatively well with what Michelle had seen in her field study and what they saw in their lab column study. Right away after we put that manure on, we tend to see more antibiotic resistant genes available in that soil for transport. With time, that tends to decrease. What's really interesting about this study is when we irradiated the soil first, all of a sudden we got much, much larger amounts of antibiotic resistant genes in the soil, right? Since there wasn't bacteria already there, the bacteria added with the manure had little to no competition, got to proliferate and spread their genes far and wide, right? So what that tends to indicate is that if we have a relatively robust, healthy soil that has a reasonable microbial community in it, it will be harder for the microbial community added with the manure to persist. They will die out more quickly with time. And that's what we want, right? If the antibiotic resistant genes are associated with things that we're adding with the manure, if there's already more competition there, they can't find space and they get out competed and die off, it helps remove those genes. Switching topics a little bit, certainly we know that if it's present in the manure, we put it out in the soil, the growing conditions in any year greatly impact how many of those genes actually move. Some work that Michelle's done up at Nashua shows that in drier years, the bacteria associated with manure and their antibiotic resistant genes don't move to tile water nearly as much. Not a surprise, right? If water isn't making it down to that tile line, if there's nothing to move those bacteria, they're not moving on their own, right? So it is very weather dependent. But in those wet years where some drivers are present, and since most of our microbial communities in manure tend to like it a little bit wet, right? They've lived in wet environments their whole life and we've selected for that. They tend to last a little bit longer in those years and be more likely to move. So if that stuff is present in our manure, we put it out in our field, it becomes very weather dependent, whether it moves or not. One thing that we could think about as ways to sort of mitigate that is, are there treatments that we could do beforehand? This is a study where basically we have two treatments. They have that original manure, so it's DO, and they measured the log number of colony forming units per gram of sample. And in this case, that is a liquid manure, and they did it for both erythromycin resistant bacteria, so counting the number of bacteria that grew on a plate with a certain concentration of erythromycin on it, and then they did the same thing for tetracycline. And then they looked at two different treatments. What happens if we're actively composting that treatment? So they took some liquid swine manure, about 3 kilograms of it, mixed it in with some hardwood sawdust, uh, added a little water to get to optimum moisture conditions, and then performed a composting process where it was heated up to 55 degrees Celsius. And then aerobically composted. They kept it aerobic by moving air through it, versus a lagoon treatment system where it was that same swine manure, but kept in a bottle at lab temperature. And then they did aerate the headspace to keep it aerobic, but they weren't aerating the manure. So big differences between the lagoon and the composting are both the temperature. The composting is much warmer, 55 degrees Celsius compared to lab temperature, which would be somewhere around 20 degrees Celsius or 70 Fahrenheit. So the composting was a little more than 2.5 times the temperature, so quite a bit warmer. And then the amount of air that it got. So the composting was kept in very aerobic conditions. The lagoon was kept in sort of an anaerobic condition. In both cases, they saw a significant decrease for the composting, right? So we saw large decreases in the number of colony forming units that were able to persist after 48 days. They chose 48 days in this case because that was essentially the time it took the compost to finish. With the tetracycline, that got some of their samples down to basically non-detect limits, not quite as effective at treating the erythromycin resistant, but they did see some efficacy there. With lagoons treatment, where it was kept sort of anaerobic on a lab bench, again, they used 48 days. Typically with the lagoon, we'll probably have, or a manure storage, we tend to have a much larger retention time than maybe that 48 days. In Iowa, we're probably looking at 6 months, so other treatment might be a little short. But for that tetracycline resistant gene in the bacteria, with just that lagoon treatment, they didn't see any difference. They did see a small but statistically significant decrease for the erythromycin resistant bacteria. That was all done with plating, which is a nice way to do it. Certainly, it's very clear. See if it grows, count it. But there are a lot of people working specifically on genes. So one of the ways that you can do this is run PCR and basically count the number of genes. I'm showing this data basically to show that it mirrors pretty similar what they saw for actual resistance when they plated, right? Which is nice. You want to see the two things correspond reasonably well. In this case, they do have another sampling point there, that 17-day window, and they chose that one basically because that would be the initial heat time of a compost pile, right? So they were trying to see, is it that initial heating, or do we have to get to stabilization with composting to reach that sort of steady state? And you see pretty similar results. In this case, they looked at two genes associated with tetracycline, so tetracycline M and tetracycline O. There's a large number of genes out there that people say are the ones you should look at. This paper actually looked at something like 6 tetracycline genes and 6 thymucin genes. I'm not going to show you all of them because they do correspond relatively well to what we saw with their actual plate counts in all cases. A follow-up study, this one I wanted to talk about, it does start with dairy manure, which, all right, is a little bit different. We don't have as many dairies here in Iowa, but what was interesting about it is they did look at 4 different treatments. So they have basically untreated, just stored manure, anaerobically digested manure, where it was in a completely mixed digester running at roughly a 30-day hydraulic retention time uh, at 35 degrees Celsius, so roughly 100 degrees Fahrenheit. Uh, Composted manure, and then the dewatered solid manure. And they tried to look at both the genes right after treatment. How did the treatment impact that, as well as follow it through both land application, looking at it for it in soil, and then they grew a root crop, right? This is how you know it's not an Iowa paper. They grew carrots in their soil to see if any of those genes made it from the soil, from the manure into the soil, and then eventually into the carrots. So would it impact either the food that we're eating or food that we're feeding to livestock? This first table here that I showed you is the impact that that treatment, either anaerobic digestion, dewatering, or composting had on indicator and pathogenic bacteria. So there's a reasonably long history of trying to use some of these treatments to get rid of pathogens. In all cases, we can see, especially with the anaerobic digestion and the composting, it did have some impact on what was left, right? So when we look at that anaerobic digestion, while total coliforms were reduced by roughly half, for many of those other either fecal coliforms or indicator bacteria that are potentially pathogenic, we got down below detection limit. For composting, we tended to see the same thing, where many of the compounds moved down towards either detection limit or below detection limit. With dewatering, there tended to not be much of a change. Uh, Potentially lots of reasons for that, right? The biggest one being most of the time when we're dewatering, it's just a partitioning or separation sort of system. And depending on how effective your system is, some of those pathogens or bacteria could be ending up with the solids, right? So if we're spinning it hard enough in a centrifuge, we can capture those solids. If you're not separating the small solids where the bacteria are most prevalent, you might not be separating them at all. So it is really dependent on how good your separation tends to be, on how effective that is. I know Michelle and I have done and are doing some more work looking at what speeds we'd actually have to do to separate those bacteria in different fractions. More importantly, for what we're talking about today, is what does that do for antimicrobial resistant genes? And you might think at first glance, well, I expect them to do the same thing, right? If it's killing out pathogens, it probably removes antimicrobial resistant genes. Unfortunately, that isn't always the case, right? Because the antimicrobial resistant genes aren't always associated with pathogens. So they might act a little bit differently. On this graph, what we're looking at here is if it is bolded, right? So if we see this bold letter or bold item, that would mean that doing that treatment process, in this case, anaerobically digesting the manure, statistically increased the prevalence of that gene, right? So that bacteria grew and tended to be more higher in concentration after treatment. If we see it's underlined, that would mean that it is statistically significantly lower in concentration. So for both the digested manure and the dewatering manure, they tended to have very little effect on antimicrobial resistant genes via those treatments. On the other hand, composting that manure, we see that most of those compounds where we see anaerobic resistant genes tended to be statistically reduced via that treatment. I do want to caution that anaerobic digestion alone, composting alone, manure treatment alone aren't the only ways to tackle this problem. And we see some research here, which I did a poor job putting the key in there. So this red circle and all the red dots indicate pigs that were administered an antibiotic and what the prevalence of their gene, that antibiotic resistant gene, was in their feces. So the red circle would be basically our control group. They didn't receive any antibiotic. That was sort of the microbial community antibiotic resistant gene threshold to start with. This green dots represent individual pig guts where they were injected with the antibiotic at a prescribed rate. And the green circle would represent the bacteria, at least on average, that we expect to generate from that. You can see that there's a shift from the red to the green, indicating that that treatment does cause some increase in antibiotic resistance prevalence in the feces from our pigs. On the other hand, this blue circle is when that antibiotic was added to the feed, right? So rather than doing a specific injection to a pig, they were feeding it in the feed. And what they found is that, maybe not surprisingly, running that antimicrobial product through the pig's gut caused a larger change in the prevalence of antibiotic resistant genes in our in the pig's feces relative to when we did injection. And they hypothesize that that's because it causes a bigger disruption in the bacterial community present in the pig's gut, right? So if we're feeding it through the pig, we get a larger change and saw more of those genes when then they were doing specific injections, at least on average, because their biggest change actually is one grain point right there where they did an injection. But I point that out because while manure management certainly can play a role in this, many other people, veterinarians specifically, are trying to look at this from a cause side of how do we treat pigs to make sure we're maintaining health while still trying to minimize some of the prevalence of antibiotic resistant genes. So where do we go from here? Certainly, this is a topic where there's a lot of active research and a lot that we still don't know or trying to do better. Many people are trying to look at, is it possible to reduce in-farm antibiotic-resistant gene proliferation by doing different treatments that improve farm management? For example, looking at different animal diets where maybe we can, for whatever reason, get some of the health effects of treating with low levels of antibiotics without actually adding them to the diet, or changing our antibiotic use strategies, either specific injections to pigs or better management of pig health through potentially precision ag tools for specific monitoring of animals. On the manure side, certainly an area of active research is looking at, are there manure treatments that we could be looking at that can help get rid of some of these antibiotic resistant genes. In some cases, that means moving towards a more municipality style wastewater treatment system where we're doing some sort of disinfection to remove it. Certainly those can be relatively costly. So one of the areas that people are trying to really work on and focus on is, are there ways to do this that are maybe going to be cost effective for farms to actually implement? And then an area where I think there's a lot of work to do and people are trying to conduct data-driven research is, just because these genes are present, what does that mean for how it impacts human health, right? We can measure whether the genes are present in the manure, whether they stay present in the soil after manure application is done, but it is really hard, and people are actively trying to work on this of what does that actually mean for making human drugs less effective. Just because they're there, does that mean that we have a problem? And that's an area where I think we're going to continue to see a lot of the heat and debate about what does this really mean and what should animal agriculture be doing. I didn't talk about that really at all today because we're focusing on the science of what do we know works, what do we need to find out if it still works. Uh, as a side note, there is a current project at Iowa State. It is led by Dr. Adina Howe, looking at, does the use of antibiotics and the type of antibiotics influence antimicrobial resistance in manure at all? And specifically, they're going to try and get manure samples from barns with and without antibiotics in the feed with different treatment strategies to see if they can link in-barn management to the prevalence of those genes. With that, they're going to look at some of those treatments that we saw with the dairy manure, trying to focus in on swine manure, composting, some anaerobic digestion and solid separation to see if there are ways to partition them and remove them from the manure. And one thing that's going to be a little bit different from their study compared to some of the ones we looked at there is rather than stopping at treatment, they're going to try and follow that through with land application. For instance, composting looked relatively effective at removing antimicrobial resistant genes from the manure prior to application, but we don't really know if that changes how prevalent they would be in the soil, right? The soil is an aerobic condition. Maybe we don't see as much die-off following application of that material as we would in the untreated manure, or if that manure had been run through an anaerobic process where it goes from anaerobic in the treatment to aerobic in the soil. So hopefully that will give us some more information specific to Iowa soil conditions and Iowa specific manure treatment practices to understand what issues we might face in the future. So with that, we'd be happy to try and take any questions or discussion you might have. Otherwise, thank you for joining us today. I don't see any questions, which is perfectly okay. If you do, feel free to contact either myself or Adina Howe, and we will do our best at answering and addressing them. We do hope to do this again next month in a day in September. NRCS is working on a new 590 standard. I Haven't heard if it's approved yet. If it is, we're hoping to have them come and talk through some of the changes they are seeing in it. If it's not approved yet, I'll see if I can get them to talk about what some of the potential changes they are looking at and how it could impact manure planning here in Iowa. So I think that should be an exciting topic for all of us next time. If not, I promise that we'll find something equally enthralling to work through. So thank you all for joining us today. It was great having you.